This video demonstrates how to do Play-Doh mitosis. First, some cell parts. Here in blue is the cytoplasm of the cell. We have the centrioles, and they help with cell division. And then we have the nucleus. Outside, covering the whole thing, is a nuclear envelope. Then we have the DNA, which we call chromatin in this case, because it is all mixed up like this. We can also say that it is unwound. In the very center of the nucleus, we have the nucleolus, which is a darker section, and it also is where ribosomes are made. First, we have interphase, and during interphase, the cells grow in size. And we can't see it, but inside the nucleus, the DNA is being copied. So there are now 2 copies of each chromosome inside. Now we start cell division, also called mitosis. First we have prophase, and during prophase, the centrioles start to move towards opposite ends of the cell, also called poles. As they separate, there are spindle fibers in between them. So they keep moving, and the spindle fibers keep growing. I'm going to move the nucleus aside for a second. And what I'm doing is I'm making dots in lines. These spindle fibers are going to connect to each chromosome and help pull it one way or the other in the cell. Also during prophase, the nucleolus disappears. The nuclear envelope breaks up and disappears. And the chromatin condenses, which means it coils up, and when it coils up, it gets thicker, and we are actually able to see at that point chromosomes. So I have some chromosomes already made, so I'm going to switch these out. And your cell needs to have at least 3 chromosomes in it. The next phase is metaphase. During metaphase, the chromosomes will be pulled by the spindle fibers so that they line up in the middle of the cell. So if you pretend that the centrioles are the North Pole and the South Pole, right here at the equator is where we have our chromosomes lining up. Metaphase means meet in the middle. Next we have anaphase, and during anaphase, each sister chromatid, or chromosome, gets separated. Remember that we actually have 2 copies here. So when they get pulled apart, they kind of look like V's, and one part is going to eventually go over to this side of the cell, and the other part will come over here. So first we separate them all, and when I'm tearing them apart, I am tearing them apart at the center, which is called the centromere. Okay, so the spindle fibers are going to pull these chromosomes over towards the centriole. These are going to get pulled this way. Okay, and that is the end of anaphase. Finally, we have telophase. And at that point, we don't see any spindle fibers anymore. So we're taking out those little dots, getting rid of them. The reason we don't see them once again is they were the ones that pulled the chromosomes towards the centrioles. Next, we have a new nucleus begin to form. So what happens is the chromosomes unwind again and go back to being chromatin. Still DNA, it's just all mixed up where we can't really tell where individual chromosomes are. So that happens on both sides. A new nuclear envelope forms around each group. New nucleolus becomes visible. And then the final step is cytokinesis, which means the division of the cytoplasm. And it just starts to curve in, and what we have here is what we call a cleavage furrow, and it finally totally breaks off and separates. Now we have two identical daughter cells, and each one will go back into interphase until it's time for mitosis to happen again.